already completed prokaryotic cell. Now, today we'll start on eukaryotic cell. So, eukaryotic cells, they are these membrane-bound. Eukaryotic cells have these membrane-bound organelles that prokaryotes didn't have a nuclear membrane that eukaryotes do. And eukaryote, this name came because "eu" means true and karyote means that they have a true nucleus. That's why eukaryote name came. So eukaryotes, they have, like, we are now talking about what the eukaryotes have. So first, let's discuss about cell wall. So among the eukaryotes, some protists and all fungi and animals have cell wall. So, sorry, among protists, all fungi and plants have cell wall. So that is cell wall absent in animals. And also not all protists, only some protists have cell walls. So these cell walls are not identical to these organisms. They have different composition. However, if fungi, the cell wall contains polysaccharide called chitin. So mostly the cell wall is most made up of cellulose. Some are made up of chitin. What are chitin? They are polysaccharide containing nitrogen. So this chitin is very similar to the exoskeletons of insects. I mean, how the insects exoskeleton are very hard, it's very difficult to break. That rigidity, that same thing, it's because of chitin that is present in this fungi. That some fungi have this chitin in their cell wall. It is similar to the skeleton of insects. Now comes cytoplasmic membrane. Cytoplasmic membrane sorry, connection issue. Cytoplasmic membrane, they're also called plasma membrane or cell membrane. Also called or cell membrane. Okay, so they are semi-permeable membrane that separates the inside of the cell from the outside. So its structure and composition are in eukaryotic cell. The plasma membrane consists of proteins, carbohydrates, and two layers of phospholipids. Phospholipids are lipid. Okay. But this is an important point. Eukaryotic cells contain many membrane-bound organelles that carry out specific cellular process. This thing is absent in prokaryotic cells. They do not have membrane-bound organelles. This only prokaryotes have. This only eukaryotes have, which prokaryotes don't have. So the chief organelles, first is nucleus. The nucleus, they 90% of the cell's DNA, and it contains the nucleus, it contains 90% of the cell's DNA. Contains 95% of the cell's DNA. So the structure, it has a nuclear envelope. It's a double membrane with nuclear pores, and that surrounds the nucleus. And it has chromosomes. Okay, the chromosomes are the genetic material that composed of DNA and the associated chromosomes, DNA and others. The chromosomes are linear. So the function of the nuclear is to carry the carrier of the hereditary information, which exerts a continuing influence over ongoing activity of the cell through protein synthesis. So they are the control center of the eukaryotic cell. This is an important term, control center of the cell. So everything, all the information which should be passed on to the next generation, everything is there in this nucleus. Okay, so the function of this nucleus is it isolates the DNA in eukaryotic cells. So here it's saying nuclear envelope. It's a double membrane with nuclear pores that surrounds the nuclear. So this nuclear pores are, or nuclear pore complexes, they are embedded in the nuclear envelope. These complex structures, they control the movement of protein and the nucleic acid, and it's the RNAs across the nuclear envelope. So they're embedded and this control the movement of this control the movement of proteins and RNAs across the nuclear envelope. So, and second comes mitochondria. So mitochondrion, we all know it's called the powerhouse of the cell. So why it's called? First, let's see the structure. So usually it is a oval shaped double membrane. It has smooth outer membrane and a folded inner membrane. So this inner membrane, that the foldings, the folds provide more surface area of, and the surface area is needed for chemical reaction to take place. The more folding will be there, the more surface area they will get, and the more it will give a site for the more chemical reaction. So the function of this mitochondria is they break down. Why it's called the powerhouse? They break down energy containing organic molecules that are the carbohydrates. Okay, so they break down the carbohydrates and repackage the energy into smaller units. There's our ATP. ATP is adenosine triphosphate. So they break down carbohydrates and the energy units they make into smaller units, ATP. These ATP are used by the cell. That's why they are the powerhouse of the cell. So mitochondria, there are two types. I mean, not two types. We can divide it in outer mitochondrial membrane and inner mitochondrial membrane. So what does it do? The outer mitochondrial membrane, it consists mostly of phospholipid and it contains a considerable amount of cholesterol. Consists mostly of phospholipid and cholesterol. Where the inner mitochondrial membrane, it is very rich in proteins. I'm writing it in a different color. Now, endoplasmic reticulum. Endoplasmic reticulum of two types, smooth reticulum and rough endoplasmic reticulum, denoted as SER and RER. So the structure, I mean, they're interconnecting flattened sacs, tubes, and channels. And their types are they're both types, both this smooth and rough type. They support the cytoplasm and they provide more surface area inside the cell for chemical reactions to take place. Actually the main function of this endoplasmic reticulum is, I mean, these organelles, they are involved in protein synthesis, transport, modification, storage, and secretion. They do a lot of things. So here, first, let's the rough endoplasmic reticulums. Ribosomes are attached to it. Ribosomes are attached to it. Okay, they are the initial modification of proteins happens here in the rough endoplasmic reticulum. And how does it happen? The polypeptide chains are formed at the ribosome and some of them are transported into the rough endoplasmic reticulum for modification. The polypeptides are then packed in transport vesicles or sacs. Okay, a piece of that endoplasmic reticulums. Okay, that pink that goes around the polypeptide. Then these vesicles transport the polypeptides to the Golgi complex for further modification into proteins. So the proteins, that the polypeptide chains, first they will form at the ribosome. Then they will transport it. They are transported in the rough endoplasmic reticulums. From there, when modification, after modification, they will be packed and then they are moved further to Golgi complex for the further processing. Whereas in smooth endoplasmic reticulum, no ribosomes are attached. Says on this point, no ribosomes are attached. They are the main site of lipid synthesis. Rough endoplasmic reticulum was for protein synthesis, the smooth endoplasmic reticulum are the main site of lipid synthesis. Lipid, again, from both of the endoplasmic reticulums, the final product is sent to the Golgi apparatus or the Golgi body. The lipids are then sent to the Golgi body in transport vesicles for further modification and distribution. So whatever is happening in the endoplasmic reticulum, it will be transported to the Golgi bodies for further modification. So the endoplasmic reticulums. Let's summarize this. Smooth endoplasmic reticulum, rough endoplasmic reticulum. The smooth one, it is smooth. Why it is smooth? Due to lack of ribosomes. So they are smooth due to lack of ribosomes. If they are smooth, then that means rough due to presence of the ribosomes. Second point, smooth, lipid, the main side of lipid synthesis. Side of synthesis. Remember it like this. Like lipid means fat. What is the first thing that comes in your mind when you think of fat? Butter. Butter is smooth. So smooth endoplasmic reticulum are for lipid synthesis. Okay. And rough endoplasmic reticulum. This should be one. Two. Side of protein synthesis. Like protein are, their textures are not that smooth, they're rough, and those people who go to gym, they look very rough and tough sort of. So you can remember it like that. That protein are associated with roughness. When butter is smooth, butter is fat, lipid. So smooth endoplasmic reticulum, they are the main site of lipid synthesis. Whereas rough endoplasmic reticulum are the main site of protein synthesis. Now, Golgi complex. Okay, Golgi complex or Golgi apparatus, what you can say. So they are the structure. There are 4 to 8 flattened membrane brown sacs. They loosely stack on top of one another. They loosely stack on top of one another, surrounded by vesicles. they're flattened sacks and they're stacked on top of one another. They look like a sack of pancakes mounted on each other. So the function of these are final modification of proteins and lipids. So this Golgi complex, important site for the formation of lipoproteins. So this each eukaryotic cell, they contains a unique stack of smooth surfaced compartments or the cystine. They're called cisternae that make up the Golgi complex. Okay. This cystine it I'm writing it here. Cystine. Okay, these sister E, they make up the Golgi complex. Okay, what are these? They are smooth, unique stack of surface compartments. Okay, so this 4 to 8 flatter membrane from sacks loosely stack on top of one another. These are cisternae. Their technical term is cisternae. They make up the Golgi complex. Now, lysosomes. Lysosomes are cell organelles found in cell which contain packet of enzymes. In this, the enzymes. Why these enzymes are important? These enzymes, the enzyme that these lysosomes contain, they are like for breaking down of proteins, lipids, etc. We need these enzymes. So lysosomes contain enzymes for breaking down proteins, lipids, etc. So, this breaking down of proteins and lipids, the digestion is within the cell itself. Okay. Then ribosomes. So they may be free in the cytoplasm or they're attached to the rough endoplasmic reticulum and the nucleus. In the rough endoplasmic reticulums, there are ribosomes, but they can be freely moving, free in the cytoplasm or it's attached to the nucleus. This same arrangement was there for the ribosomes attaching to the nucleus. We have seen it in the prokaryotes. So ribosomes These structures are not bound by a membrane. These structures are not membrane bound. And they are called means these structures are not membrane bound and they are made up of RNA and protein. Okay, so these ribosomes are called protein factories since they are the main site of protein synthesis. See their function: major sites of protein and polypeptide synthesis. The structure are called protein factories because it's the function main. They are the main site of protein synthesis. Now, these polypeptides, they are the chains of amino acid. Chains of amino acid. Now, in prokaryotes, we have studied that the ribosomes are of 70s, of 50s, and 30s. But here, the eukaryotic ribosomes are of 80s. How 80s? 60s is the larger subunit, where 40s is a smaller subunit. And now the last, cytoskeleton. Cytoskeleton, the structure, by structure, you can say they are a network of filamentous protein structures called microtubules, microfilaments, and microtrabeculum. This is cytoskeleton. Let's divide this. It's already written microfilaments. Now, what are the functions? Okay, these microtubules, they provide internal structure to the cell and helps in maintenance of shape of eukaryotic cell. They provide internal structure to the cell and helps maintenance of shape Eukaryotics. Then comes microfilaments. By name, you can say that they're filaments. They they are more slender and cylinder-like structure. I mean, how filaments look? They're like cylinder-like structure. They're made up of this contractile protein called actin. Made of actin. So they are linked to the inner surface of the plasma membrane. They're made up of actin. Plasma membrane. And these structures are involved in the generation of forces for internal cell motion. Okay. Generate forces for motion. So whatever happens within the cell, the motion, that is because of these microfilaments. They are they are the one that generate the force. The last one is microtubule. So they appear to be very fragile tubes. Okay, but they form a transient network in the systole. Systole. Sorry, cystosome. Cystosome. So what are cystosomes? They are, I mean, the fluid portion of the cytoplasm, exclusive of all the organelles and membranes. Those are cystosomes. This organelles-free sap is called cystosome. Okay. Exclusive of organelles membranes. Okay. It's the fluid portion exclusive of organelles and membranes. So this is a prokaryotic cell and this is an eukaryotic cell. I mean, I've put these two pictures side by side and you see and compare how complex the eukaryotic cell is as compared with the prokaryotic cell. They don't have much organelles. And they're not very complex either. I mean, all the organelles are like they're put together. They're put together. They don't do much, but the eukaryotic cells are very complex. I mean, see the eukaryotic cell. Everybody has the eukaryotic cell. A cell that a fungi has, the same cell that a human has. A mammal, any type of mammal, any plants, animals, everybody has the same. So the more these are the complex living organisms, so therefore eukaryotic cell is more complex than that of prokaryotic cell. Now there are so many, eukaryotes. From the eukaryotes, we'll discuss about the basic yeast and molds. We'll discuss about these two because from the food microbiology point of view, these are important. So this is are eukaryotic microorganisms. They're classified in the kingdom fungi. And this yeasts are unicellular. Although some species of yeast forms may become multicellular through the formation of string-connected budding cell known as pseudohyphae or false hyphae, as seen in most molds. It will be like this. Yes. And this type of branching will appear. These are not actually hyphae, but like this budding, they won't. This budding, when they will be that budding happens, and when they will be attached to it, they become multicellular. Mostly, it's visible in molds and some yeasts. Now, the size of the yeast it can vary greatly depending on the species. Typically, measuring 3 to 4 micrometer in diameter, although some yeast can reach over 40 micrometer. Just see 3 to 4 to 40. I mean, they are like 10 times more. Most yeast reproduce asexually by mitosis, and many do so by asymmetric division called budding. So mitosis, mitosis, meiosis and mitosis. In mitosis, what happens? A cell, it replicates its chromosomes. First, it will replicate its chromosome. Like this will be the cell. Then they will replicate the chromosome within the nucleus. Then two identical, then a cell will replicate the chromosomes and then it will segregate them. This is how it happens. See, first the nucleus will be separated, then the cytoplasm, and then two daughter cells will be. So, kids, its chromosomes, and then so this what happened to so here two identical nuclei is formed. Two identical nuclei. It will be in the preparation for cell division. Two identical nuclei will be there and then it will be separate. Where in budding, we all know, suppose it's a yeast cell. Then a bud will develop like this, elongation. Then a new bud. See, two different nucleuses formed. Here you can see the elongation of the nucleus is also taking place as well as the cytoplasm. So by fermentation, next point, by fermentation, the yeast species Saccharomyces cerevisiae. It's very common. Saccharomyces cerevisiae, what it does, it converts the carbohydrate to carbon dioxide and alcohol and then it is, yeah, should be full stop. Huh. So the yeast species Saccharomyces cerevisiae, it converts the carbohydrates to carbon dioxide and alcohol. So this is a very old practice, all right. For thousands of years, the carbon dioxide has been used in baking and the alcohol in alcoholic beverages. So this yeast is an extremely important as a model organism in the cell biology research, and it's one of the most how can I say, like one of the most researched, not like thoroughly researched eukaryotic microorganisms. So this first, what happened? First, researchers have used this yeast to gather information about biology of the eukaryotic cell, and then to human biology. There are other species of yeast and like some pathogen which cause infection to the humans. So this yeast is it's very much as an field of interest in research. So yeast so their nutrition and growth. Yeast are chemoorganotrophs, means they use organic compound as a source of energy. They use organic compounds as a source of energy and do not require sunlight to grow. Remember, yeast doesn't need, do not require sunlight to grow. Not require sunlight to grow. So how does, carbon is important for most the living beings. So how does this happen? The carbon is obtained mostly from hexose sugar, means six carbon sugar, hexose sugar like glucose and fructose, or disaccharides such as sucrose and maltose. So here some species can metabolize pentose sugar like ribose, alcohols, and organic acid. So this yeast species, they either require oxygen or aerobic cellular for aerobic cellular respiration, or they're obligate anaerobes. That means, so yeast cells are obligate anaerobes, but they also have aerobic methods for energy production. They are also facultative anaerobes. So yeast grow best under aerobic conditions. Okay, but also they have for energy production, they respire aerobically, so they are also called facultative anaerobes. Yeast grow best in neutral or slightly acidic pH environment. Then yeast vary in what temperature range they grow best. There we can't say, means there are exceptions. For example, Leucosporidium frigidum. It's a type of yeast. It grows at -2 to 20 degrees Celsius. Okay, very low temperature. It's an exception. Generally, the optimum, this is not a common range. Then Saccharomyces cerevisiae at 5 to 35 degrees Celsius. Sorry, Saccharomyces telluris. Saccharomyces telluris is 5 to 35 degrees Celsius. Candida slofi at 28 to 45 degrees Celsius. So we can see that the range never lives more than 45. So the maximum, if we generalize this, the Yeast optimum temperature can say in general 25 to 30 degrees Celsius, whereas maximum temperature that they can withstand 35 to 47 degrees Celsius. This is the highest limit they can have. So these cells, they can survive under freezing, survive freezing under certain conditions, but their viability, I mean their usefulness, it will decrease over time. They can survive, but like slowly, they will lose their viability. So in general, yeasts are grown in lab on solid growth media or in liquid growths. So we use selective agar for yeast. Like in nutritive or this type of agar, we cannot grow. So the common media used for cultivation of yeast include potato dextrose agar. It is the most common agar, potato dextrose agar or potato dextrose broth, both. This is the most common, mostly in labs, you will see. Then Wolfstein Laboratories nutrient. WLN agar, yeast peptone dextrose agar. This is also very common in industry level or in school laboratories. You can find. Then yeast mol agar or BOT, YM. These are the common selective media for yeast and mold. So the appearance of a white thready yeast, commonly known as calm yeast, this name is important. Calm yeast is often a byproduct of lactofermentation, that is pickling. During pickling of certain vegetables, this calm yeast is produced. Its appearance is white and it's a thready type of yeast. And why it does? It is usually the result of exposure to air. When this yeast happens due to exposure of air, but it is harmless, but it can give a bad flavor. Okay, and that's why it's not desirable. I mean, it's not pathogenic, it's harmless, but it gives a bad flavor to that pickled vegetable, and it should be removed during fermentation and must be removed regularly during fermentation. Now, reproduction, like yeast, like all fungi, may be asexual and sexual reproductive cycles we've discussed. So the most common mode of vegetative growth is in yeast is asexual reproduction by bud. So we discussed this here. A small bud, or also known as flap, or a daughter cell is formed on the parent cell. Now the nucleus of the parent cell, it splits into the daughter's nucleus and migrates into the daughter cell. And this elongation happens. This is the cell. Both the nucleus and the cytoplasm will start to elongate. The bud continues to grow until it separates from the parent cell, forming a new cell. But here, the daughter cell produced during budding process is generally smaller than the mother cell. Now, some yeast, what they do, they produce they produce by fission, therefore creating two identical sized daughter cells. This is a difference. During fission, that is in the first we discussed how it happens. During fission, the two daughter cells will have identical size. They have everything equal. But it won't be in budding. In budding, the size of the daughter cell will be smaller than that of the parent cell. Now in industry, Industrial application will discuss about it elaborately in the next chapters, but just an overview. So this useful physiological properties of yeast, they like that's why we use it. In they're very much used in this industry because of this useful this properties of yeast. What they do, they can ferment the sugar. So this fermentation of sugar by yeast is very old, and it's one of the largest application of yeast in this in the technology. So many type of yeast are used. We know about baker's yeast. It's for bread production. Then there are brewer's yeast, alcohol like beer production, beer fermentation. Then yeast in wine fermentation. For xylitol production. So there's many types of yeast are used for this. So first, let's talk about alcoholic beverages. So this alcoholic beverages, they contain ethanol. Okay. Then how does ethanol? Ethanol is produced by fermentation. So under anaerobic, in alcoholic beverages, the production of ethanol by fermentation. It's done by yeast. Production of ethanol by fermentation. So this the carbohydrates by certain species of yeast under anaerobic or low oxygen condition, it happens. Now some of that are beer, like the beer. They when we ferment the yeast at low temperature, we get the beer. Okay, so this brewing yeast, they can be classified as top cropping or bottom cropping. So some for beer production, there are some like Saccharomyces pastorinus. This is useful for the beer fermentation. This is in use. So in wine also, I mean, what does in beer there are oat, that they ferment that thing. What happens in wine? I mean, actually, beer making is a like huge process. I mean, I can't explain it to you right now in one or two lines. In when we will be studying about the uses of microorganisms in this food products, we will get a detailed idea. But I mean, just this name is very common, Saccharomyces pastorinus. This is for beer fermentation. And in wine also, we need that. What happens? It converts the sugar present in the must or in grape juice into ethanol. So yeast is normally already present in the grape skins. So I mean, like this, it done. Now for baking, the most common one is Saccharomyces cerevisiae. It's used baking is in baking as a leavening agent. Okay, where it converts the fermentable sugar present in the dough into the gas carbon dioxide. Okay, in baking, converts sugar carbon dioxide. This is very common. So this causes the dough to expand. I mean, whenever for bread making, after like adding yeast or more, we leave them for some time and then they expand. This conversion of sugar into carbon dioxide, this causes the dough to expand or rise, and there will be gas form pockets or bubbles we can see in the dough. And this is common like bioremediation. What is in bioremediation? What we do, we use the living organism for the removal of that for removal of toxins or pollutants or contaminants. With the help of living organism, we do that. This is bioremediation. So some yeast have the potential for it. Okay, one is Yarrow, Yarrowia lighting. What it does, degrade palm oil, milk, effluent. So this is it degrades the that in oil meal, especially in palm oil meal, the effluent it degrades that. I mean it before leaving it to the environment. If we degrade it, then it will cause less harm. It won't spread pollution, and also TNT. TNT is an explosive material, and other there are other hydrocarbons such as alkanes, fatty acids, fats and oil, definitely. So for bioremediation, this type of, this is very common in like now people are working on this. They use nanotechnology and different other things. This type of for how to control, how to remove the toxins and the pollutants which already present in the environment. So this name is very important. Don't forget that. And for industrial ethanol production, yeast is number one choice. This ability of yeast to convert sugar into ethanol, it is means very. By biotechnology industry for ethanol fuel, it is very much harnessed by the technologies in this industry. So. So what Saccharomyces yeast have been genetically engineered for this purpose to ferment xylose, one of the major fermentable sugar that is present in this biomass. So like that they produce ethanol and they later use it as a fuel. So these are the application, industrial application. For yeast. Then we'll study about this as mainly the food products in later chapters. Now mold. This term mold, it's given by the appearance only. I mean the form and the structure of mold. That anything we call it mold. The term mold is very common to means that the thing which we see in the whole mass that we see in top of the food when it is spoiled, the name it got like that. So the molds are fungi in the form of multicellular filaments called hyphae. So a network of this tubular branched hyphae called a mycelium is considered a single organism. In the this hyphae is generally transparent. So this the mycelium, what it does, it appears to be very fine, very fluffy white threads. It will be there all over the surface, but it won't look like it's a single cell because what we assume single cell to be very small and I mean a whole, but there are branches in the mycelium and it is considered as a single organism. Uh, so these molds they play a very important role in biotechnology and food science in the production of various foods and beverages. Like this, this is some common. I mean, there are so many, but these are some common. Like the Koji molds, they are a group of Aspergillus species, notably Aspergillus oryzae and A. sojai. They have been cultured in the Eastern Asia for many centuries. Why? Because they are used to ferment soybean and wheat mixture to make soybean paste and soya sauce like that. So they are very common in countries like Japan and like other countries that those are present in the East Asia, Korea, like that. So this koji molds, they break down the starch in sorry, the koji molds, they are there to ferment soybean and this for they ferment soybean to make soybean paste and soy sauce. Now, what the koji mold does, they break down the starch in rice, barley, sweet potatoes, etc. This process is called stratification. Saccharification. This process is called saccharification. So the saccharification it is for the production of sake, soju, and other distilled spirits. For this we need this. What is sake? They are fermented rice, and these are like sake is an Japanese alcoholic drink. Soju is a Korean alcoholic drink. I mean, this type of drinks are made up of these koji molds. So these koji molds are used in the preparation of also it's a Japanese dish. Katsuobushi is a type of tuna. Okay. Also, red rice yeast is a product of the mold Monascus purpurus. It's grown on rice and it's very common in Asian diet. The yeast contains several compounds collectively known as monocolins, which are known to inhibit cholesterol synthesis. So these are some important points. There are, other points also there. Like for the production of cheese, we use that mold ripened cheese. The mold ripened cheese. Some cheese like blue cheese, Roquefort, Camembert, Brie. These are mold ripened cheese. We need molds for them. Like Geotrichum candidum, we need this. We will read about this when we study the fermentation and the production of food products by microorganism. So these are very like common right now. Now this Eastern Asian culture is so much is involved. With this type of questions may come, like questions about NATO. When in 2000, I forgot the year. At that time some NATO questions came. Like they're very famous Japanese products, and also focus on this type of food products and try to remember these names. Okay, so our unit 1 is completed. Thank you.